for first, second, and third best paper awards are there, and with Professor Shantanu Pal, co-judges with Dr. Obik Kumar Chaudhary from Vivekananda Centenary College, Rohora, and Professor Shantanu Pal belongs to Department of Botany, University of Calcutta. So participants are requested to arrange their posters according to the serial number as sent to you. So our first, without going to any delay, our first presenter, can I request to our first presenter, PP1, Pratiksha Mishra, and she will be presenting effect of carbon-nitrogen ratio on polyhydroxybutyrate accumulation by a mangrove isolated cyanobacteria, Limnothrix sp. Pratiksha, you can share your presentation. Pratiksha Mishra, you are the first presenter. Hello, Pratiksha. So we go to our next presenter, PP2, Karishma Das, and she will be presenting algicidal activity of associated bacteria alkali genes, Aquatilis on Lingbia asutari, and its effect on growth and cell viability of cyanobacteria. Karishma Das, please share your presentation.
Yes, sir.
This is PP2.
Okay.
Hello.
Hello,
So one thing, can you hear me? So one thing is that you make your presentation by 8 minutes. Okay.
Yes. You will get 8 minutes for your presentation, and 2 minutes or 3 minutes for discussion. Okay.
Okay, sir.
Okay.
So actually, the sharing option I cannot found.
You just click the share screen in the green color. In the bottom.
Yes, bottom yes, yes, sir. Can it be visible, sir?
Yes.
Yes, sir. A very good afternoon to everyone. I'm Karishma Das, and I'm a PhD scholar of Utkal University, Bhubaneswar. And my topic is algicidal activity of associated bacteria, alkaligenesis equatilis on Lingbia estuary and its effect on growth and cell viability of cyanobacteria. So we know that on the topic, about the topic, if I am saying, we know that both cyanobacteria and bacteria are the organism which we found in the natural habitat, in aquatic habitat and in terrestrial habitat, and both are the prokaryotes. Then what is the interaction between the cyanobacteria and bacteria? We know both are prokaryotes, both have similar characteristics, but cyanobacteria is photosynthetic. And what is the interaction between bacteria and cyanobacteria, and how bacteria inhibit or increase the growth of cyanobacteria? If we want to increase some algal mass, or if we want some algal mass harvesting, then we use many chemical compounds, but by using the biotic compounds, by using the living things like bacteria, we can increase the mass of algae. Also, we can control the harmful algal bloom also. So my topic is based on the subject about the interaction between cyanobacteria and bacteria. The bacteria is associated with that cyanobacteria, and I have selected my samples from the mangrove area of Bhitarkanika from Odisha region. My bacteria is Alkaligenesis equatilis, which was found in the association of Lingbia estuary. And the whole topic about the negative interaction between the bacteria and cyanobacteria, we know they both live in the natural habitat, and they both show some types of relationship. The relationship may of commensalism, the relationship may of parasitism, the relationship may of the amensalism also. And in my study, the relationship between my bacteria and hello.
Yes, go
Yes,
on.
Hello.
yes, you are audible.
Yes,
Audible,
sir.
audible.
Yes, sir. And in my topic, the bacteria and cyanobacteria, they they do not help each other. The bacteria inhibit the growth of cyanobacteria. Then the first three things we want to know. We need to know the first three things. For the first one thing is, what is the interaction between the mechanism? What is the mechanism between the mechanism of the interaction between cyanobacteria and bacteria? Then why bacteria inhibits the growth of cyanobacteria? Then what is the application of the negative interaction between bacteria and cyanobacteria? In my study, in my poster, you can clearly see that in my past culture, when we compare the 0-day and 18-day culture, the 0-day culture, I have inoculated same amount of C, that is exenic cyanobacteria, and CB, that is co-cultured cyanobacteria, means the Lingbaia estuary with alkaligenesis equatilis. In the 0-day, from the 0-day to the 18 days, I have cultured my exenic cyanobacteria and cyanobacteria under co-cultured condition, and you can clearly see in 18 days the growth of exenic cyanobacteria is more than that of the co-cultured condition. It means bacteria secretes some things. Bacteria secretes some hormones, some enzymes, some amino acids, and some inhibitory product which inhibit the growth of cyanobacteria. So in the plate culture, you can also see in the when we compare the 0-day and 18-day culture, you can also see in the 18-day culture, the growth of cyanobacteria plus bacteria is less than that of the exenic condition. It means that in exenic condition, the cyanobacteria can grow more than that of the co-cultured condition. Then these are the visualization of in flask culture and in plate culture. Then I have done some autofluorescence capacity, and I have done some microscopic view. I have observed some microscopic view under fluorescence microscope to observe the fluorescence capacity of my cyanobacteria under exenic condition and co-culture condition. In 0-day, you can see that the cyanobacteria under exenic and co-culture condition show same amount of same fluorescence capacity, but when the day goes on, it means when the 0-day to 6-day, then 9 days, then 12 days, then after 18 days, you can see here when we compare the 12 day and 9 days, we can compare the 9-day and 12-day condition. The exenic condition show more fluorescence capacity than that of the co-culture condition. Here in the co-culture condition, you can see some breakdown of cells. Some the cells are getting darker than that of the exenic condition, and so overall, under fluorescence microscope or under the visualization in flask culture and plate culture, there is a decline in growth. There is a decline in the fluorescence capacity. There is a decline in growth under the co-cultured condition than that of the exenic condition. And you can see same thing when I have observed that exenic and co-culture condition under the microscope with trypan glue. We know that trypan glue is used to detect the dead cell, and when the trypan glue is observed by the cells, and when the dead cells, the cells are more dead, they observe more trypan glue. Then when in 18 days, you can see there is clear dead cells. The whole image is like looking like a blue color. The clear dead cell in that of the co-culture condition. So in co-culture condition, bacteria helps in the lysis of some algal cells, helps in the death of some algal cells. So here I can say that my bacteria is an antagonist bacteria. There are two types of relationship. One is the synergistic and another is the antagonistic. So here my bacteria plays in an antagonistic role in the cyanobacteria, in the growth of cyanobacteria. And why this is happening? Why my bacteria inhibit? What is the secret? What are the secretory things? What is the secretory things which they secrete to inhibit the growth? And here is a scanning electron microscopic image. You can see that in the exenic condition, the cells are so smooth. The cells are looking like so smooth, so silk. But in the co-cultured condition, the cells become rough. The cells become rough because the bacteria degrades some cellular structure of the cyanobacteria. And here I am giving some dry wet. The blue graph is the exenic condition, and the red graph is the co-culture condition. So here in the 6 days of incubation, the cyanobacteria plus bacteria, the cyanobacteria under the co-culture condition on the 6 days of incubation, it is increased. Then after 6 days, the stationary phase comes. But if we compare the exenic and co-culture condition, there is increase in the growth of exenic condition than that of the co-culture. So I am saying in a layman language that that if someone do not know anything about the cyanobacteria and bacteria interaction, they will say me that they are found in the pond. Then why you are saying about their interaction? I can say that they both are prokaryotic. We live in a society. We meet many people. We meet many type of people. Some are our friends. Some are our enemies. Some we do not like. Some we like. So here there are many type of bacteria. There are many type of cyanobacteria, but their relationship are of different type. Some bacteria helps to increase the growth of cyanobacteria. Some bacteria are a very good friend of cyanobacteria, and some bacteria inhibit the growth of cyanobacteria. And they both antagonistic and synergistic application. The synergistic interaction have different application. So here in the growth condition, in the dry wet condition, you can see a increase in the growth of axenic, that of the aquaculture condition. And if I wanted to know about the reason of the decline in growth, then what are the reason of the decline in growth? My bacteria produces something which helps to decline the growth of cyanobacteria. My bacteria produce cellulase. You can see in the plate culture the cellulase producing bacteria, which in the CMC agar media I have cultured it in the CMC agar media. So my bacteria Alkaligenesis equatilis is able to inhibit cellulase and it is able to produce cellulase and protease, which inhibit the protein and cellulose of my algal cell, which may be the reason of decline in growth. So here in the picture you can see the clear inhibition zone cellulose producing and protease producing, which are the main reason of the growth inhibition. And there is another image which is the urease production and bacteriocin production. Bacteria produces many type of toxic compound which are antimicrobial compound which are harmful for the algal blooms. And I found my bacteria Alkaligenesis equatilis was able to produce the bacteriocin, which is a type of toxin, and I have cultured on the MRS media. You can see the clear inhibition zone that it produces bacteriocin, which acts as the algicidal compound. Bacteriocin is an algicidal compound, which may be another reason of the decline in growth of the cyanobacteria. And it is also capable for the production of urease. Urease is also an inhibitory compound. There are many inhibitory compounds like phenol, tannin, alkaloid, which are also produced by my bacteria Alkaligenesis equatilis, which may be the another reasons of in decline in the growth of cyanobacteria. And here is the phylogenetic sequencing of my cyanobacteria and bacteria. My cyanobacteria was identified as Lingaya ascheri, and my bacteria was identified as Alkaligenesis equatilis. Here in a cartoonic image, I can say you clearly that there are two types of interaction I have given. One is synergistic, another is antagonistic. Here the bacteria is very much happy because it gives indoleacetic acid, vitamin B12 and siderophore, and the cyanobacteria take it, and in exchange of this beneficiary compound, in exchange of this growth promoting compound, the cyanobacteria gives some nutrient to the bacteria. You know that both bacteria and cyanobacteria live together. We live together in a family. Our father gives us gives us the financial support. Our mother gives us food. Then they are both beneficiary to each other. The cyanobacteria gives some nutrient to bacteria. The bacteria gives some IA, vitamin B12, siderophore to cyanobacteria, because cyanobacteria are unable to produce this, but cyanobacteria need this for their growth. So they are both benefiting each other in their growth. But in the second image, you can see the cyanobacteria is crying because bacteria gives tannin, phenol, alkaloid, toxin, urease, etc. Yes, sir. The conclusion is what are the application of the cyanobacteria and bacteria negative interaction. There are many harmful algal blooms which produces many algal toxin in the ponds, and we can give our bacteria, we can apply our bacteria to control those harmful algal blooms. We can give those bacteria, we can apply those bacteria for the wastewater treatment, because we can control the harmful algal blooms, the cyanotoxin produced in the ponds. We can use it for the wastewater treatments like pollutant removal, like the toxicant removal, like a microalgal harvesting. So there are many applications of synergistic and antagonistic interaction of cyanobacteria and bacteria, like microalgal harvesting, pollutant removal, harmful algal bloom control, wastewater treatments, etc. So in my future purpose, the future purpose of my study is the interaction between cyanobacteria and bacteria. I have observed this cellular interaction, this growth interaction, this protease cellulase, this interaction. But I want to study about the metabolic interaction, the genomic study, the proteomic study. Is there any exchange of gene or protein compounds between the bacteria and cyanobacteria, which is also helpful in the decline or increase in the growth of cyanobacteria? So in my future study, I want to study about the metabolic, proteomic, genomic study in the interaction. What is the physiology between cyanobacteria and bacteria interaction in the natural habitat? Thank you, sir.
Thank you, Karishma. Is there any question regarding this poster? You may ask any one of the participants or judges.
Yes, participants, first you ask. It's open for discussion.
Okay, there is a question in a chat box. Okay, there is a question for Karishma. But what if the bacteria is harmful for human being?
Yes, sir. There are many bacteria which are harmful for the human being, but we can apply those bacteria only to control the harmful algal bloom. If we apply the bacteria to control the harmful algal bloom in a water, then we can later we can also apply we can apply some antimicrobial compounds to control those bacteria also. But our aim is to control the harmful algal bloom, which produces some toxins, some antibacterial, some compounds, some toxic compounds, some cyanotoxins. So our main aim is to control the algal bloom. Then if we want to control the algal blooms, we can also control the bacteria by applying some antimicrobial compounds.
So Karishma,
yes, sir.
It's a good word, but what is the amount of bacteria cyanobacteria you have taken in your culture?
Yes, sir. Actually, my amount is 1:1. I have experimented different ratio: 1:1, 1:2, 1:3, 3:1, and 2:1. But the best result was given is the 1:1 ratio. So I have yes, sir.
Okay. But what is in nature? In nature, what is the amount? What is the ratio we find in nature?
So in the
because you are actually mimicking what is in the nature, no? Yes. Bacteria, cyanobacteria. So you have to have an idea what is the ratio of cyanobacteria to bacteria or bacteria to cyanobacteria. Got my point? To
yes, sir,
originally mimic.
Yes, sir. Yes,
yes. You got a good result in this ratio and you're showing that ratio. Okay.
Yes, sir, yes,
that may not be true. You got my point?
Yes,
because
yes,
you
sir.
do some application study. You don't want to just pinpoint your result, but maybe in this soil or in the environment, that ratio does not work out.
Okay, sir.
I don't have any idea. But you have to find out what is the ratio, what are the different ratios that can be found in the nature. And that you have to experimentally prove.
Yes, sir. Yes.
In your in vitro setting. Okay. That is one thing. So, so there are many bacteria, those are known for producing cellulase. What is so special about this?
Sir, no, sir. Actually, this bacteria is associated with the cyanobacteria, with my cyanobacteria. That bacteria was associated with my cyanobacteria. It was not different. When I have isolated my cyanobacteria and I have cultured on it a plate culture, then that particular bacteria was associated with my cyanobacteria. So I found that in the natural habitat that Alkaligenesis equatilis was also associated with Limbaya. Then what is the interaction between those two that they are found in association with each other? What is the mechanism of interaction? This is not about only interaction. This is about the associative interaction. What is the interaction between bacteria and its associated?
But you can also think of isolating if there is cellulase in the culture. You can isolate the cellulase. Okay.
Yes, sir. Yes.
And another thing is that it's not only this bacteria and cyanobacteria. There are many other microorganisms that are present there. No.
Yes, sir. Yes, sir.
So in the environment, when you want to mimic an environment, that is very difficult.
Yes.
We're just putting two parts and we don't have the known ratio even there.
Yes.
Of the nature. You got my point? So I
Yes,
think
sir.
you take care of those things. Okay, thank you.
Okay, sir. Thank you.
Okay. There are two more questions from another judge. First question is, what are the outgroups in the phylogenetic trees, and what bootstrap value is taken for the phylogenetic tree?
So the phylogenetic tree was neighbor joining phylogenetic tree.
And what bootstrap value is taken for the phylogenetic tree?
So
Is there any bootstrap
value,
value?
bootstrap value actually I have done that in a mega X software, and I've done counter jokes, I think this type of name, this type model.
Okay, okay. You have used a specific software or any?
Yes, the mega X software.
Okay, okay, okay. So is there any more questions? So we can proceed our next presenter. Pratiksha, are you there? PP1. Pratiksha, I just have seen Pratiksha. I don't know what happened to Pratiksha.
So you go to the next one.
Next, okay, PP3, Manjurima Ganguly from Guru Nanak Institute of Pharmaceutical Sciences
Hello,
and Technology,
hello.
West Bengal.
Sir, am
Yes,
I
you are
audible?
audible, Manjurima. And she's going to present wastewater treatment using microalgae, eco-friendly option. Okay, time starts now, Manjurima.
Okay,
You
sir.
can share
Is it
your
visible my screen?
Yes, yes, yes. This is visible.
Okay, thank you. Thank you for firstly, good evening, everyone, and thank you for the amazing opportunity to present my review paper along with my teammates Pooja Haldar, Debolina Chakraborty, and Dr. Bhaskar Chaudhary on wastewater treatment using microalgae, eco-friendly option. So in India, as we know, it is a developing country. Due to increased population and excessive industrialization, the amount of wastewater that is being generated is very high. So there needs a measure that needs to be taken. Basically, that is what explains our review paper. So, due to this, water pollution is one of the most critical environmental problems we are facing each day. So, for wastewater treatment, various conventional methods we use. But in India, these are not very cost friendly and economical. So, nowadays, some new green technical methods are evolving, and microalgae is one of them. From the last 50 years, biological wastewater treatment systems with microalgae have gained importance, and now it is widely accepted, and it is a conventional method of wastewater treatment. So, cultivation of microalgae in wastewater is basically production of energy from microbial biomass, and it is a very common technique. In this diagram, we can see the basic operation principles for the microalgae production combination with wastewater treatment. Basically, microalgae have the ability to remove nutrients, heavy metals, organic and inorganic toxic substances and other impurities present in the wastewater by using the sunlight, carbon dioxide and various other nutrients. The main advantage of using algal system is that it absorbs the solar radiation in form of energy in chloroplast cells and takes carbon dioxide along with nutrients from wastewater to synthesize their biomass and produce oxygen. Now, the oxygen that is released from microalgae is enough to meet the most aerobic bacterial requirements while metabolizing the residual organics in the treated wastewater. So, algal compounds that can be assimilated in aqueous system. The bacteria in turn constitute an essential source of carbon dioxide required for algal growth, stimulate the release of its vitamins and organic growth factors, and change the pH, supporting the medium for algal growth. So now, sir, we can see that there are various factors that affect these growth of microalgae in the wastewater. As for example, the sunlight. It is an essential thing for photosynthesis, and without photosynthesis, microalgae will definitely not be able to grow in the wastewater. So it is very essential. Next comes the carbon dioxide. So this microalgae utilizes carbon dioxide from the atmosphere
as carbon source to produce oxygen. Now studies have actually proved that microalgae are more efficient in fixing carbon dioxide than actual terrestrial plants. So the greatest advantage of microalgae are the nutrients. So municipal and industrial wastewater, they have abundant amount of nutrients and suspended solid materials in them. So microalgae utilize them for photosynthesis and actually helps in forming the cleaner water. And the biomass that is actually here in the diagram, we can see the biomass that is left after the wastewater treatment. It can also be used in various other sources like biofertilizers for animal feeding and so on. So while actually reviewing this very interesting subject, we have come across the works of various famous environmentologists. So let's just see that what were the observations. I in this review paper have mentioned about 3 very important review of literature. One is Chatterjee et al. So she in 2013 proved that algal production systems are very efficient in nutrient-rich wastewater stream, and after 14 days of continuous batch culture, it was observed that 94% of ammonia, 89% of 89% of TNPT was removed by algae. So another very notable observation in this was by Wong et al. In 2010. So in this study, various microorganisms proved to be very effective in nutrient removal by microalgae. In this diagram also, I have noted in this table some microalgal species and their wastewater characteristics like chlorella and Nuzena. It settled domestic sewage and 92, 74, 97% of biological oxygen demand, 87% of chemical oxygen demand was actually decreasing within the retention time of 10 hours. Few other example of such algae are cyanobacteria, chlorella vulgaris, chlorella pyrenosa, and so on. So various other such studies include studies of Mahapatra et al., which was actually held in Mysore, and 82% of biological oxygen demand, carbon oxygen demand, was actually removed from the wastewater. Now, why are we actually using this method of microalgae wastewater system and not the conventional usual method of microalgae system? There must be some reasons because of that. So I have tried to note down few of them. As for example, in case of conventional conventional removal of waste from water, there is no advantageous biomass that is left. But in case of recovery of nutrients from microalgae, there are certain valuable biomass which can be used as fertilizers, animal feed, and so on. Apart from that, reduction of carbon dioxide emission in the environment is a very important factor. It actually decreases the pathogens in a very cost-effective way, which is not possible in the conventional method we use. Another very important factor is algal system absorbs solar radiation and take it in chloroplast cell and utilizes the energy it produces from photosynthesis for the main work basically. Henceforth, it is like the greatest advantage, and it is very cost effective. So to conclude, we can say that microalgae have the potential to uptake carbon, nitrogen, and phosphorus and other heavy metals that is there in the wastewater in a very cost-friendly and economic way. Sewage and industrial wastewater is naturally enriched in nutrients that can be used for algal growth. It is also observed that a major factor that affecting algal growth and treatment efficiency are carbon dioxide and light from all other factors. The various studies we actually discussed about conducted to treat the wastewater using microalgae shows that this reactor that we use has a significant reduction in nutrients, BOD and COD and other toxic chemicals, but also it has a certain increase in the total suspended solute in the wastewater. So that is why it is very necessary to actually remove the microalgae from the treated effluent to meet the general standards of wastewater discharge. So the nutrients removal efficiency of microalgae based on wastewater treatment system is very high. The system has a removal efficiency of approximately 78 to 99% of nitrogen and phosphorus and 40 to 65% of COD, BOD respectively and other impurities present in water. So Sir, that's what I wanted to present for this seminar. Thank you everyone for the opportunity. Thank you.
Thank you, Manjarima. Now the is open for question and answer.
So Manjarima, so this is a review work, isn't it? You have done a
Yes,
review
sir.
work.
Yes, sir. We have tried to review the various work which were already done on this topic and tried to accumulate the data for further research work and what are the advantages.
So
And
one of
presented
the
it to
things,
paper on it.
so one of the thing, how do you, what is the mechanism? You have read a lot. So in a nutshell, how microalgae, what is the mechanism of microalgae for the treatment of waste material?
Right, sir. So basically what, and the mechanism is also the greatest advantage of microalgae. Basically it uses the energy from sunlight, it uses the atmospheric nitrogen, carbon dioxide and other nutrients. And basically it utilizes that for photosynthesis. And the nutrients that are already present in the wastewater, it also utilizes that and prepares, and basically utilization of this energy actually helps in removal of and reduction of the BOD and COD and removal of the wastewater.
No, I was thinking there, if you have any idea about, if you look at the chemistry, okay, so there is some chemistry going on there. It's biology and chemistry together. So is there any role of some metal, non-metal? Do you have any idea how microalgae, what is the mechanism as far as chemistry is concerned? It's basically a chemistry, okay? It's not magic. You are seeing the end product. You are seeing the first product as microalgae. Then end product is that. But in between the system, there are some chemical reactions.
Right, sir. Yes, sir. As I said, it can actually has the efficiency to absorb the nutrients and in the atmospheric gases and also the metals. So I think that is what is
So
the
what
main
are the
mechanism
metals, do you think? What are the prominent metals, do you think, that are removed by this microalgae?
prominent metals that are present in that actually helps in the eutrophication of the wastewater system. It helps in the removal of those, basically.
You got my point. I was just thinking of, you are just looking at the biological aspect, the first thing and the end thing, but in between this thing, there are a huge chemistry going on. It's basically organic chemistry going on. So what are the things going on? You need to know that. Okay.
Right, right. Okay.
Any question from?
Any question? Okay, there is another question. What to do with the biomass once used for the treatment of water? Can it be reused? How?
Yes, it was also mentioned with the diagram in my poster presentation. The biomass that are produced after the microalgae wastewater production, it can definitely be reused. It can be reused as biofertilizers. It can be also used as animal feedings and everything. That is how it can be used.
Okay, I hope there is no more question.
I'm sorry, I was actually muted. So was my answer audible? The last answer.
You can say it again.
Okay, I'm really sorry. Yes, the biomass that is produced, it can be reused. It can be reused as biofertilizers, or it can also be used for feeding for animals and other cattle. It can be used in various environmental factors like that.
Okay, that means there is a biorefinery way.
Right.
I have a question.
Okay.
Am I audible?
Yes, yes, sir.
Yes.
So how do you distinguish between, say, natural water and wastewater? Is there any criteria for that?
So difference between wastewater and natural water, of course, there are certain criteria for that. constituents of a natural water is very much different. The chemical constituents and everything in a natural water is very much different from a wastewater basically. There are harmful and
So how is
hazardous
it
elements.
different? So how is it different? Is there any specific criteria? You have mentioned about BOD, COD, all these things. So is there any specific criteria or reference scale by which you can categorize that this is wastewater and this is not?
Sorry, can you just repeat your question? It just got very. I'm really sorry, I couldn't actually hear what you were saying.
Yes, actually, since yours is a theoretical work mostly, so there are specific let down criteria. Okay, so which one is in wastewater? Those has been proposed by the Central Pollution Control Board. So if you go to their website, then you can get all this information.
Right, sir. So basically, BOD or biological oxygen demand basically represents the amount of dissolved oxygen consumed by the biological organisms when they decompose organic matter in water. So right, and the chemical oxygen demand is the amount of oxygen consumed when the water sample is chemically
Right.
oxidized, right?
So if the BOD is less than 1 milligram per liter, then that is put under category C. So that then becomes
right.
wastewater. So if it is more than 1, then it is
Right,
not a
sir,
wastewater. So those criteria you should know about.
Not
Okay.
wastewater. Right, sir. Right, right, sir. Right, sir.
Thank you, Dr. Chaudhary. Okay, thank you, Manjurima. Now we can go to our next presenter. Pratiksha, are you ready?
Yes, sir. I am audible.
Okay, you are audible. You can share
Thank you,
your
sir.
screen. Yes. This is PP1, first poster, PP1. And Pratiksha will be presenting effect of carbon-nitrogen ratio on polyhydroxybutyrate accumulation by a mangrove isolated cyanobacteria, Limnothrix.
Hello.
Okay, you may present, Pratiksha.
Hello. Good afternoon, everyone.
If you need, you can zoom in. Okay.
Okay. Hello.
Yes, you are audible.
Plastic have been an essential part of the modern human lifestyle since their discovery. Unfortunately, their non-biodegradability is a significant disadvantage. The longevity causes accumulation in the terrestrial and marine ecosphere. Considering this strain, biodegradable and renewable alternative to conventional practice are becoming increasingly important. A promising class of bioplastic is polyhydroxyalkanite, which has the similar property to our commercial plastics. And an alternative approach is the cultivation of phototrophic microorganisms such as cyanobacteria for the bioplastic production. Cyanobacteria use oxygenic photosynthesis, which employ atmospheric carbon dioxide and light as energy source. PHB in cyanobacteria is mainly built up from glycogen and other carbon storage from synthesized carbon storage forms synthesized during nutrient deficient condition. Cyanobacteria produce much lower level PHB during their phototrophic conditions. So different neutral optimization conditions are preferred for the increasing the polyhydroxybutyrate concentration in the cyanobacteria. In this study, in this work, Limnothrix plantonica is isolated from mangrove region of Odisha, was tested under phototropic and mesotrophic condition with various carbon and nitrogen ratio to determine its potential for the PHB accumulation. The strain was grown in ASN medium with various concentration of carbon sources like glucose, acetate, fructose, lactose, starch, and sucrose. From these carbon sources, acetate is the base carbon sources with and without nitrogen. Low PHB concentration were present at the beginning in the mesotrophic stage in all cultures. Supplementation with sodium acetate and nitrogen depleted, PHB content increased a maximum to threefold than the phototropic condition. At their stationary phase, the highest PHB concentration was observed after 9 days in the mesotropy along with the nutrient limitation. The PHB content was increased remarkably 21.3 microgram per liter, a threefold rise as compared to the control. Subsequently, the concentration decreased with increasing the time period after ninth day. Acetate accelerated the PHB formation and ensured the PHB increment within short time. Although the PHB content is 10.9 microgram per liter in N deficit condition, which is greater than phototropic condition. Therefore, an N limitation is beneficial for large scale application. This study investigated the PHB formation in 3-stage cultivation process containing a growing stage, micronutrient deplicated phototropic stage, and a subsequent mesotropic stage. The cultivation in nitrogen deficiency and supplemented with acetate result in the increasing of 184% of PHB than control. Hello? Hello?
Yes, yes, you are audible.
Audible.
Thank you, sir.
Okay, you completed your presentation?
Yes, sir. Yes, sir.
Okay, okay. Is there any question you may ask from the participants or from the judges? Pratiksha, which one is better, polyhydroxyalkanoate or butyrate obtained from algae? Which
So
perform better?
polyhydroxybutyrate.
Because bacteria is also producing both.
Yes, sir. But bacteria is a heterotrophic organism and require very much carbon sources. But cyanobacteria is a photosynthetic microorganism and can utilize atmospheric carbon dioxide for PHB production.
Okay. So they can fix natural carbon dioxide. That means they can fix natural carbon into
Yes,
carbon
sir.
compound. Okay. Is there any more question? So we otherwise can move to our next presenter. That is Pallavi Ghosh.
Sir.
Pallavi Ghosh is presenting use of algae to sequester heavy metals, a phycoremediation approach. This is PP4. Okay, fourth presentation, PP4.
এই আমি বড় করতে
Okay.
পারছি না।
Pallavi, share your screen.
Yes, sir. I am sharing. Sir, is it visible?
Not yet. Yes, it is visible now.
Okay, sir. So, good afternoon, sir. Today, my topic is use of algae to sequester heavy metals, a phycoremediation approach. So, phycoremediation is basically a type of bioremediation where microalgae and macroalgae are used to remove pollutants and xenobiotic from wastewater and CO2 from waste air. So, heavy metals are released into the environment by natural processes like flood, wind, volcanic eruption, and anthropogenic activities like industrial effluents. Heavy metals are non-biodegradable in nature and very much toxic to plant, animals, and overall the ecosystem health. There are some techniques like leaching, hydrolysis, polymer, microencapsulation, chemical extraction, precipitation, ion exchange, and electrolytic technologies that are used for the removal of heavy metals from polluted area. But there are also some major problems in these techniques that they are either very much expensive or sometimes less effective for large-scale projects. Algae is a simple, non-flowering and typically aquatic plant for a large group that include the seaweeds and many single cell formed. Algae contain chlorophyll but lack of true stems, roots, leaves and vascular tissues. Algae play a major role in aquatic ecosystem by constituting the energy base of the food chain for all the aquatic fauna. As an autotrophic organism, algae transform the H2O and CO2 to sugar by the process of photosynthesis. When plants and animals take up such contaminated water, heavy metals, that is HM, may accumulate into the water, into them. Where from there, they may enter human food chains. To remove HM, a variety of remediation procedures have been used. In the past, a variety of traditional methods like ion exchange, electrolytic technologies, chemical extraction have been used. However, the bulk of these methods are not cost effective for large scale projects and require strict supervision and ongoing monitoring. Additionally, these methods are not very good in getting rid of heavy metals. Algae are a sustainable and environmentally beneficial option for removing heavy metals from contaminated water in this context. Phytoremediation benefits from the buildup of heavy metals by micro and macroalgae compared to other methods that are not economical and not environmentally friendly. The main sources of heavy metal, their harmful effects on people, the potential of algae in phytoremediation, and their methods of heavy metal uptake are all covered in this review. And here is the mechanism. In this photo, we can explain the algae have a good binding affinity towards nutrients like nitrogen and phosphorus and toxic compounds like heavy metals and dyes because of various functional groups on its cell surface by following the mechanism such as biosorption, bioaccumulation, or alternate biodegradation pathway. After attaching with the functional group, the algae secrete the biodegradation enzyme, which can easily degrade toxic complex compound into simple non-toxic compounds. Now we can get the pure treated water, and further we can also use this treated algal biomass for feedstock for bioenergy production and animal feed. This total procedure in this chart, this total procedure is done by primary, secondary, and tertiary process. In primary remediation, there is a form of sedimentation. In secondary, activated sludge is formed, and after that, the tertiary, where the again sedimentation and filtration happens. In this chart, we will discuss about the various types of algae used to remove the heavy metals like Glyocapsa species, Chlorella vulgaris, etc. Now is the advantages. Algae have the ability to remove diverse contaminants like heavy metals, pesticides, inorganic and organic toxins. They are cultures of microalgae may be cultivated in large-scale reservoirs, open ponds, and also in the laboratory. Sustainable and eco-friendly from an ecological perspective. Possible co-production of biofuels and biofertilizers. Flexible to handle fluctuation and in quality and quantity of effluent feed. Numerous species of algae have been discovered as viable option for the removal or detoxification of heavy metals and as prospective, cost-effective alternative to physiochemical remediation methods. The majority of microalgae have numerous mechanisms for securing heavy metals ions, making them identified as potential bioabsorbent. Numerous studies support both their superiority over a number of alternative physiochemical and conventional approaches, as well as their value in the large-scale cleanup of wastewater. Some cutting-edge solutions, such as genetic alteration that increase biomass output, may be able to overcome these issues. Additionally, a practical and long-lasting approach for deciding on the best physical condition and bioabsorbent, as well as for pinpointing the main issue, need to be established. But in order to create a healthier environment, it is crucial to take into account a number of microalgae remediation techniques. Through the use of genetic engineering, it is possible to improve the development of transgenic organisms that overexpress phytochelatins and methylothioneins, which can bind to heavy metals and transport them into vacuoles, facilitating phycoaccumulation and the removal of hazardous elements from aquatic habitats. These are all the references from where we make this poster. And lastly, we will thank to our director Abhijit Sengupta sir and principal Professor Lopamudra Datta for giving us the opportunity to present our presentation, and also grateful to our mentor Dr. Bhaskar Chaudhary for assisting us in this webinar presentation. And thanks to all my fellow mates who helped me to make this poster. And also thank you, sir, to give me this opportunity to present here. Thank you so much, sir.
Thank you, Pallavi. Any question from the audience? Any questions? I think. Dr. Chaudhary?
Yes, I have a question that what are the functional that are involved in this bioabsorption process? There was no information on this in the presentation. So obviously the algae has certain functional groups and the metals either remain in an anionic or cationic state. So what are the main functional groups that are involved?
Sir, I can't hear you. The CH functional group, sir?
Yes, so these are either anions or cations, right? What is getting assimilated in as heavy metals? So they are in the ionic states, right?
Sir, these are cations. Hello?
All of these are cations. None of them are anions.
Sir, anions can
Yes.
also be
Am I
there.
audible?
Yes, sir. Anions can also be there.
Yes. Okay, so what are the functional groups on the algal surface which are inverse? There has to be some binding between the algal surface and metal. So what are the functional groups?
Sir,
Where do these metals bind before it gets into the system?
Actually, the question is what are the functional groups present on the surface of the algae which may interact, I think.
COH group, sir.
COH.
COH
NH2
or
group.
COOH?
COO, NH2 groups.
Okay.
These are the groups present in the form of the compound.
Okay. Another thing, while you present any poster, make sure your scientific names are all correct. Okay. So, as for example, you have written reticulate, it's reticulata. Okay. So that last A is missing in your presentation. So make sure the scientific names are correct.
Okay, okay, sir. Actually, there may be some typing mistake. Okay, sir.
Yes. Yes. Okay.
Thank you, sir. Thank you so much.
Thank you, Dr. Chaudhary, and thank you, Pallavi, for nice presentation. And next presenter, PP5, Suparna Sharma from Guwahati University, and she is presenting phytoplankton diversity in a brick-clean waste drain of Mandia region. The time is yours, Suparna.
Yes, sir. So is my screen visible?
No, not yet.
So is it visible now?
Yes. Yes, now it is visible.
Okay.
Just
So good afternoon, everyone.
Yes.
My topic for today's presentation is phytoplankton diversity in bricklin waste strain of Mandya region, Borpeta, Assam. Moving on to the introduction part, phytoplankton communities are important in environmental impact studies because of their extreme sensitivity to change conditions. Phytoplankton play a major role in the biosynthesis of organic matters in aquatic bodies. Bricklin industries play a major role in unorganized sector of the economy in India and are restricted mostly to rural and semi-urban areas. These industries profoundly affect the surrounding environment by conversion of wetlands and agricultural lands into degraded wastelands. The impact on the aquatic bodies, particularly the plankton communities, have been less studied in this area, and therefore the present study was undertaken to study the planktonic composition in aquatic bodies near bricklin industries. The collection of sample was done for a short period of time for the month of February and March of this year, and the collected samples were collected from the site. The site of the photo of the site has been presented here. Then the samples were observed under the microscope, and after the taking proper photographs, the identification of collected algae were done. Using different standard literatures. Moving on to the result part. A total of 5 different phylums were found in that area. The dominant was Charophyta, followed by Chlorophyta, then Euglenozoa, Bacillariophyta, and Cyanobacteria. 7 palmar genera were also found, that is Enchiodesmus, Clostridium, Euglena, Gomphoneuma, Liposynclis, Ficus, and Scaradesmus. A total of 21 species were found to be new to the northeast region. Moving on to the conclusion part, altogether 43 taxa belonging to 21 genera were reported, of which 7 are palmar genera. Scaradesmus with 8 species was found to be dominant, followed by Trachylomonas with 6 species. That's all, sir. Thank you.
Okay, thank you, Suparna. Any questions?
So Suparna,
Yes, sir.
so you have done some study. So what is the uniqueness of the study? Is there any report? There is no previous report on this area or what?
Not exactly from the area from where I have collected, but there are reports from other areas. I have, while going through the literature, I found some papers related to bricklin pollution and the presence of planktonic diversity of the near and around the bricklin areas. So because the pollution from bricklin areas has been found to be at a rise. So the planktonic composition may give us an idea of the possible and potent algal composition and diversity of that area surviving in polluted regions.
So do you find any unique algae that is present in this area?
Not exactly unique, sir, but I would say algae from, I found more of green algae compared to other algae, like blue-green algae or some other algae. The number of algae belonging to green algae were more.
I think that is true for whole of Assam. Na? If you go all of Assam and see the spectrum, the green algae number is high or less or what? My thing is that whatever you have done, can you give a light on your study that this is the unique part of this study? That is what I want to know.
Okay. So as of, if I have to talk about the uniqueness, I would say as I have found more number of green algae from this polluted area, and there are also papers available based on the capability and capacity of green algae surviving in polluted areas, especially in area polluted by heavy metals, already mentioned by other presenters. So maybe that could be a uniqueness of this this area, that the presence of green algae are more in these polluted regions. So that could be one of the uniqueness, I would say.
When you do is good work, but you have to always rationalize why I'm doing this and how I can implement this in future for the betterment of society. Okay, so that's what I want.
Yes, sir.
Okay, thank you.
Okay, sir. Thank you, sir.
Okay, there is another question. What is the importance of Palmer's genera in this work?
Sir, the Palmer index shows us the intensity of pollution in a particular area.
Okay.
So the Palmer genera, those I have mentioned, are already are presented by Palmer in his paper, that if present of these particular genera of algae and particular species of these algae are present, then the pollution level may be high or organically high, or maybe more polluted or less polluted. So from my study, I have found that the site where I have collected my sample is organically polluted.
Shukanya,
Yes, sir. Hello, sir.
yes, just a suggestion. Yes, am I audible?
Yes, your
Sir,
sound
your
is
voice is
breaking.
cracking.
Yes.
Yes, sir.
Yes. So my suggestion is that in case of Palmer's index, we will see that is a calculation and there are specific range. Okay, they have given certain
Yes, sir.
range within which fall. Then you call it as polluted or non-polluted. Okay, there is a formula. Okay,
Yes, sir.
based upon the number
Yes,
of
sir.
taxa you get, whether it is green algae or euglenoids. So you have to make that calculation first and then only say that whether it is polluted or not. Simply finding the
Yes,
taxa
sir.
does not mean it is polluted. First thing. Secondly,
Okay,
try
sir.
to use more modern indices, because these are older indices, probably used around the 1970s. Okay, so there are much more modern indices which you can use, go through the references, and there are diatom specific, green algae specific. And in case of Palmer, he has only used the euglenophytes and the green algae. So if you get something other than these two divisions, you cannot use Palmer's index. So I would suggest to use more modern indices if you are doing this kind of study. Okay.
Okay, sir. Thank you so much for the suggestion.
So another question came from the audience. Have you noticed any seasonal change in algal community?
So for that, I have to go and again collect the samples. The time period was very less, actually from February to March, and after that flood occurred here in the Assam region. So therefore, I couldn't go for collection. If I do the collection for a longer period of time, then it will be possible for me to answer this question more accurately.
Right. Thank you.
Thank you, sir.
I hope there is no more question. So we can proceed our next presentation. And Nilam Jyoti Kalita, are you there? Nilam Jyoti?
Yes, sir.
Okay, so you are presenting analyzing the effects of microelement copper as a stressor in Westilopsis indica. So you can share your screen. This is PP6.
Sir, is it visible?
No, not yet. Okay, now it's visible.
Okay, now
You
it's
can
visible.
present. Yes.
You can present. Yes.
Nilam Jyoti, you are not audible.
Nilam Jyoti, you are not audible.
Please unmute yourself.
Please unmute yourself.
Shashwati Bhattacharya,
Shashwati
your presentation.
Bhattacharya, your presentation.
You can ready your
You
presentation
can ready
later.
your presentation later.
I hope there is a problem.
I hope there is a problem.
Shashwati Bhattacharya, you can present your paper.
Sir, is it visible?
Yes, yes, this is visible now and audible also. Please.
Okay, sir. Hello, sir. Good afternoon, everyone. I am Nilam Jyoti Kolita from Department of Botany, Guwahati University, and today I am going to present my work on a topic entitled Analyzing the Effects of Microelement Copper as a Stressor in Westolopsis indica. As we all know that the microelements or trace elements are the metals that are required in minute quantities for proper functioning of the cell, as well as the physical chemical properties of the body of an organism. And I have chosen the copper as a micronutrient, which is an essential element that is required for growth and development of the organisms. It is required for the development and proper biochemical functioning of photosynthetic organisms, including the cyanobacteria. Beyond the critical level, it becomes lethal and toxic, not only to the organisms, but also the whole environment. So, it's one of the major importance is that it acts as a cofactor in cyanobacteria for different metabolic processes, such as it acts as a cofactor for cytochrome oxidase, plastocyanin, etc. But in presence of higher quantities, it may be the toxic to the organisms. And I have chosen this Westolopsis indica because Westolopsis indica is one of the most dominant cyanobacteria that is found in Assam. And lots of many researchers such as Bordoloi et al. 2015, Dotto and Borua 2019, Borua et al. 2020 have discovered have isolated from different water bodies of Assam. And copper is this trace element which if it is go beyond the stress level, then it may affect the morphological senses, biochemical senses, and oxidative stress. And Westolopsis indica is a cyanobacteria which has nitrogen fixing cyanobacteria having entangled filament and barrel shaped shell, and it is a heterocystus in nature. In methodology, I have selected different ppm concentration of copper. One is control, and rest are 0.5 ppm, 1 ppm, 1.5 ppm, 2 ppm, and 2.5 ppm concentration of copper. And I have observed its different growth parameters like the growth measurement. I have followed the Odhikari et al. 2019 protocol for growth measurement For biomass estimation, I have followed the Patnik and Malik 2015 protocol. For pigment estimation, I have analyzed different pigment like the chlorophyll estimation. For chlorophyll estimation, I have followed the Lichtenthaler 1987 protocol. For phycobilipprotein, which is also a pigment of cyanobacteria, I have isolated this pigment and analyzed it. And I have followed the Bennett and Bogart 1973 protocol. And for choline estimation, which is a oxidative enzyme, I have followed the Betts et al. 1973 protocol. So moving to the result, I have found that when the copper concentration was 0.5 ppm concentration, it growth become highest, whereas from 1 to 2.5 ppm concentration growth become gradually decreases. And in biomass estimation also, I have found that over the control, the growth become highest in 0.5 ppm concentration, and there is a significant decrease of biomass was observed from 1 to 2.5 ppm concentration. In chlorophyll content, I have found that at 0.5 ppm concentration, the chlorophyll content become highest over the control, and from 1 to 2.5 ppm concentration, there is a gradual decrease of chlorophyll content was observed. In like the aforementioned, I have found that the carotenoid content was also highest in 0.5 ppm concentration over the control, and 1 to 2.5 ppm concentration, there is a gradual decrease of carotenoid content was observed. In case of phycocyanin content, which is a phycobilipprotein, one of the most important component of cyanobacteria, it was highest in 0.5 ppm concentration over the control, and in 1 to 2.5 ppm concentration, phycocyanin content become gradually decreases. In case of yellow phycocyanin content, there is a in 0.5 and 1 ppm concentration, the yellow phycocyanin content become almost similar over the similar. However, the 1.5 to 2.5 ppm concentration, the yellow phycocyanin content become decreases. In case of the proline content, the proline content become highest in 2.5 ppm concentration, and in 2.5 ppm concentration. However, the 0.5 ppm to 1 ppm concentration, the 2.2 ppm concentration, the proline content become gradually increases. So in conclusion, we can say that the copper has a deleterious effect on cyanobacteria if present in the higher concentration. But the proline production was increased with time and dose dependent manner, because proline is a antioxidative enzyme, which antioxidative component which protect the cyanobacterial cell from the stress. And the biochemical parameters such as the growth, chlorophyll A, chlorophyll B, carotenoid, and phycobilic protein contents may change along with the destruction of the cell organelles due to the effect of heavy metal stress. Thank you, everyone.
Thank you. Is there any question?
So Nilam Jyoti.
Yes,
Hello,
sir.
can you hear me?
Yes, sir.
Okay, so what is the amount of you are talking about copper? Huh? So what is the
Yes,
amount
sir.
of copper present in the soil? Do you have any idea?
Sir, sorry, sir, but I have found that in Northeast India, there is some report like the Hazarika et al. 2015 and Ahad et al. 2017 have reported that there is a presence of copper in the soil.
No, there is definitely a presence of copper in the soil. What is the amount? When you are talking that copper is giving stress. So if you give anything too much, it will give you stress, isn't it? So before you do a study, you need to know what is the amount of that thing if you go for a stress biology like that thing. So what is the amount present, okay, in the environment first? Then you can mimic that thing in your laboratory setting. You got my point? So
Yes, sir.
copper is by far the most suitable element in the human body. Okay, so it gives
Yes, sir.
much lesser stress as compared to other. You got my point? So that is the whole starting point. So if you don't know the amount present in the nature and you start giving much more or less, then it does not hold true for the assay you are doing. You got my point?
Yes, sir.
So you can give many fold, but that is not in nature. That amount is not present in nature. Then
Yes.
you cannot tell that thing. And you know in which form of copper is present in the soil? And your approach is good, but you need to start with, you need to know the concentration. Without knowing the concentration, you just don't give 1 ppm, 1.5, 2, 2.5. You first need to know what is the ppm you need to mimic. That is my question.
Okay, sir.
Because in nature, we don't face that. But if you give too much of, suppose carbon dioxide. So if there is huge amount of carbon dioxide in our room, we'll suffocate. But that is not present in nature now. So we'll have stress. So that is my point. Okay. So you need to rethink what is the amount you need to take. Okay.
Okay, sir. Yes.
Okay, there is one question in the chat box for Nilam Jyoti. What is the LC50 and LD50 for copper in the organism?
Sir, LC50 value varies in different research. In my work, if we say the LC50 value means the amount of chlorophyll content that is decreased. Any content, if we say biomass or whatever, if I consider the chlorophyll content in my work, I have found that 1.5 to 2.5 ppm, 2 ppm concentration, I have found that there is a 50% decrease of chlorophyll content over the control. That in this case, we can say that 1.5 ppm concentration is the LC50 value for my work.
I think Dr. Panchali got her answer. Okay, any more questions? Otherwise, we can go to our next presenter. That is PP7, Shashwati Bhattacharya from Guwahati University. And she will be presenting freshwater algal diversity in some riverine ponds of central Brahmaputra Valley.
Is it visible, sir?
Yes, it is visible and you are audible also. Please.
Okay, thank you, sir. Myself, Shashwati Bhattacharya from Guwahati University, Department of Botany. Now I am going to present my topic on freshwater algal diversity in some riverine ponds of central Brahmaputra Valley. First of all, I would like to say a few words about algae. We all know algae are simple, non-flowering photosynthetic plants. They are the thallophytic organisms, that is, their vegetative body cannot be differentiated into true roots, stem, and leaves. Algae are ubiquitous in distribution, ranging from marine and freshwater to desert sand, and from hot boiling spring to snow and ice. But majority of them prefer to grow in aquatic habitat. Riverine area comprises of both land and water, but characterized by limited land and land lines of communication. In other words, riverine habitat are the terrestrial zone between aquatic and terrestrial habitat. Blessed with the mighty river Brahmaputra, the state Assam comprises innumerable number of smaller to bigger ponds of different origins, which is supposed to have rich algal diversity. But from literature review, it was observed that no previous diversity work had been done from this riverine area of the Nagaon district, which lies in the central Brahmaputra valley of Assam. Therefore, the present investigation was carried out to explore the diversity from this region. This is the map of the study site. To meet the goal, a total of 20 points were selected randomly. Materials and method. Samples were collected randomly at morning hours from 5 to 9 a.m. By filtering technique using plankton net of mesh size 45 micron. Samples were immediately after the collection, samples were brought to the Plant Ecology Laboratory of Guwahati University, and samples were observed in Euromex Delphi X Observer Series microscope. Digital photographs were also taken with the help of digital camera, and the measurements were taken with the help of image view software. And the identification of algal species were done by consulting literature and monographs available in the plant taxonomy. Now moving to my results part. A total of 162 algal species were recorded under 67 genera, 38 family, 22 order, and 10 class and 7 phylum. Among them, with 47 species, Charophyta constitute the dominant genera. This figure shows the percentage-wise taxonomic coverage. Here, Charophyta constitute the dominant genera with 29%. Chlorophyta constitute second dominant genera with 27%, followed by Euglenozoa 25%, Bacillariophyta 13%, Cyanobacteria 4%, Myozoa and Acrophyta 1% respectively. Another significant result from this study is a total of 86 new reports from Assam, belonging to 29 species from Charophyta, 26 species from Euglenozoa, 16 species from Chlorophyta, 12 species from Bacillariophyta, 2 species from Acrophyta, and 2 species from Cyanobacteria. These are the photographs of some collected algal species. And that's it. Thank you, sir.
Okay. Thank you, Shashwati. Any question from the participants or from the judges?
So Shashwati have done a huge piece of work, but the thing is that if you compare, this is a part of the Assam. So if you compare the greater Assam, so the pie chart you have shown, does it matches or it is a different pie chart when you go to other side of Assam regarding the distribution of algae from Charophyta to other things? Charophyta is the prominent thing you have reported in this part of Assam. So if you take a greater Assam,
sir, from previous report, it was found that Chlorophyta was dominant, but in my result, I found that Charophyta was dominant. This is the significant result.
Okay.
Thank you, Shashwati, and thank you, Professor Paul. And now I move to the next presenter, Shobik Pramanik.
Yes, am I audible?
Yes, you are audible, and you are presenting exploration of microalgal diversity of different wetlands of West Bengal in correlation with physicochemical parameters. This is PP8.
Yes, yes.
Okay.
Hello. Am I audible?
Yes,
Audible?
yes.
Okay.
Yes.
Okay. Good afternoon, everyone. I'm Shobik Pramani. Today, I'm going to present this poster entitled Exploration of the Microalgal Diversity of Different Wetlands of West Bengal in Correlation with the Physiochemical Parameters. In this study, I had selected in total 13 wetlands from different locations of West Bengal, and samples were collecting using the random sampling methods. The collected samples were studied under light microscope and then taken from very clear photographs, which helped me to identify those organisms. The organisms are identified based on the morphological features compared with different monographs and different published papers. The physiochemical parameters were measured by measuring electrode and different titometric and calorimetric methods. In total, 42 taxa from 25 genera and 19 families were identified. As per the diversity and abundance of the microalgae, the most groups are Chlorophyceae, Euglynophyceae, and Basidiophyceae. And the genus Cenerismus saw the highest species of the diversity and was dominant throughout the study areas. And some unique taxa like Pediastrum simplex and the Planktolimbia contineria are only found in 2 specific sites. In addition, the physiochemical parameters and their correlation with the microalgal community, revealing the most of the relatively microalgal community most of the places are abundant in high relatively light intensity with high nitrate and phosphate containing wetlands. In this study, not only helped me to understand the microalgal community in these places, but also helped me to understand their salient features. And during this study, I had also isolated 5 to 6 microorganisms like microalgae like Chlorella, Chlorococcum, and Chrysmodesmus, which is subject to further molecular identifications. As per the microalgae interesting source of the nutraceuticals, I have planned to explore nutraceutical values of these isolated organisms. I am grateful to my supervisor, Dr. Ganendra, and I am thankful to organizing committee for giving me opportunity to present my work. Thank you.
Thank you, Shobik.
Thank you.
Any questions from the audience? Shobik, what you have performed statistical analysis?
CCA, canonical correspondence analysis.
Okay. So PC and CCA, any new model you have tried or not?
No. Previously models which are previously tried by others.
Okay, try some new. There are many new models. Okay.
Okay.
So
Okay. I will.
You can correlate like redundancy analysis or any others.
Okay.
You can easily correlate.
Okay.
So thank you, Shobik.
Thank you.
So we move to our next presenter, PP9, Anupam Kundu. And Anupam Kundu is going to present microalgal diversity of Ajodhya Hills, Purulia, West Bengal.
1 second. Problem হয়েছে একটু ল্যাপটপে 1 সেকেন্ড
ওকে ঠিক আছে is Orko ready or not?
Orko the and 1 second wait করুন
okay
yes sir, I'm ready. Am I audible to all of you?
Yes, you can present your poster in the meantime. So Orko is presenting, Orko Mondal is presenting PP10. Poster number is
yes
PP10.
sir. Yes sir, I'm Anupam Kundu. Yes sir. I will present my PPT after Orko Mondal.
Okay.
Thank you, sir.
You just get ready your poster.
Okay sir. Okay sir. Thank you sir.
So Orko Mondal is presenting phycoremediation potential of Nostoc SP for removal of arsenic from contaminated water. So this is PP10.
Yes,
Orko, share your screen.
Yes, sir.
Yes,
so
this is visible and audible. Yes.
Okay. Good afternoon, everyone.
You can
am I audible?
Yes, you are audible. You can share it in a full screen mode.
So this is I'm sharing.
No, you can.
So can you see this? Sir?
Yes. You can also go for full screen.
Sir, I think this is full screen. Is it not full screen to you, sir?
Okay, you present your poster. Okay.
Okay. Good afternoon, everyone. I am Orko Mondol, research scholar, Department of Botany, Vishwabharati Shantiniketan. My poster is based on phycoremediation potential of alinostoc species for removal of arsenic from contaminated water. Please allow me to tell you briefly about the background of this work. Waste material of Kolkata and surrounding are dumped in the East Kolkata wetland. Besides, there are various industries. This clearly makes it a metal contaminated area, so I have chosen this site and particularly Dhapa area for my study. Earlier, I have tested the water quality of this region and the concentration of various heavy metal collected from there was quantified. Please let me show you a little supplementary data regarding this work. Here is the table. It is to show, clearly show the arsenic concentration is higher. In the Kaneko analysis, where I found the organism like Alinostoc, Questelopsis, Formidium can grow in this arsenic contaminated area. So I took this organism Alinostoc for further in vitro experiments. So back to the main page. I also I collected the water and soil samples and concentrated the sample. Then I have documented the algal growth using the trinocular microscope and captured photographs. Then I followed various standard monograph for identification. Then next step I have included the organism into BG11 medium and also tried to prepare a culture. So I have isolated a strain of Alinostoc from this place. I have also isolated the organism and after pure culture deposited to VBCZA culture collection site, and I have carried out the molecular taxonomy study and 16S gene sequence for further confirmation. In this Bayesian inference, phylogenetic analysis of the organism where Alinostoc is close to Nostoc, and so other found to the close relation between the other organisms. I have carried out the EC50 of the arsenic tolerance with various concentration of arsenic. EC50 is where the chlorophyll is degraded to 50%. Lethal is the next higher to that, and sublethal is prior to that. In the way I have selected the concentrations. So first, this is the how I determined the EC50 value. Here you can see various concentration of arsenic, and these are the percentage of chlorophyll. It is clearly seen that there is a reduction of 50% of chlorophyll in 0.012 molar concentration. I have taken this as EC50 concentration. 0.01 and 0.14 molar concentration were selected as sublethal and lethal respectively. And all the following experiments were carried out with these three concentrations only. Next, I determined the carbohydrate, protein, and pigment concentrations. In this, the control and the 0.01 molar and 0.12 molar, and where pigment, protein, and carbohydrate is decreased in each concentration. Where I grow this organism in higher concentration of arsenic, then it was observed releasing lots of polysaccharides. So it was maybe an adaptive feature of the organism. Arsenic quantification was done by triacid digestion method, and then it was analyzed by ICP-OES. After isolation, I inoculated the organism into the selected concentration. Then I carried out the different physiological experiments. In the experiment, I have got a very remarkable data using same index and ICP-OES. I have found the index value of the same index of the samples in 3 different concentrations. I have correlated the percentages of value of same index with the dry weight of biomass and ICP-OES data. Here you can see that the EC50 concentration arsenic percentage is higher, and I have got the same data from ICP, that is 17.9 milligram per liter. Let me zoom little bit to show you. Then in case of CPS, dry weight of CPS is higher in EC50 concentration, and the arsenic absorption is also higher than the other 2 concentration, that is 11.37 milligram per liter. Dry weight of RPS is higher in the lethal concentration, that is 0.14 molar, and that is and the dry weight was 36 milligram, and the arsenic absorption is 110.5 milligram per liter in particular concentration. This has given a good result where it can be said that the production of RPS increase in response to higher metal concentration, and this RPS accumulate more arsenic. So it is a evidence. So the increase of arsenic concentration, RPS release increases, and there is a notable rise in arsenic absorption. So from this experiment, I can conclude that when alinoster grows in higher arsenic concentration, it releases more capsular polysaccharide and release polysaccharide. We can thus very easily utilize this for the phycoremediation. We can very easily extract them and separate them from the soil and water of the arsenic contaminated areas. This is really very helpful. So at the end, I can say I want to convey my heartiest gratitude to CSIR for financial support and head of the Department of Botany, Vishwabharati, for providing me the laboratory facilities. Thank you.
Thank you, Orko. Very nice presentation. And now this session is open for question and answer. Any question from the participants or from the judges?
So Orko, it's a nice word, but the thing is that alinoster, okay. So
yes,
what is the possible mechanism of absorption? Is there, have you checked on that? Why?
So what we find that only when the release polysaccharide, they grow lots of polysaccharide, it secretes lots of polysaccharide in case of higher concentration. On that basis, they absorb that metal within that polysaccharide. So that may be the adaptive feature of that organism for their survival, and in the meantime, and that is also help for the phycoremediation.
Yes, but if you go to the cellular biology part of that, is there any expression of, I don't know, receptors or any other thing that actually pulls down the arsenic inside the alinoster, or like that of thing, any receptor mediated kinetics, or it's other way, I don't know.
So first of all, we didn't go for the cellular level or molecular level. So what I find with the the ICPS, and that is the cell itself, the organism cell itself don't accumulate lots of arsenic. Only the carbohydrate parts, which they are secrets, that part they accumulate.
Okay.
Not the cellular part, that is not present.
So
Lots of
these are unique carbohydrates or what? Polysaccharides?
So that is the release polysaccharide is a compound polysaccharide. We try to identify which kind of saccharide that is. We able to identify only 3 or 5 present monosaccharide with us, but there is lots of 2 or 3 more saccharides which we need to be identified again.
So you are
that
talking
is not
about
available.
This polysaccharide has binding affinity towards arsenic.
Yes, sir.
That those are trapping the arsenic. Am I right?
Yes, sir. Exactly. Okay. Yes, sir. Exactly.
Okay.
So, any more questions? So, I may request Anupam Kundu.
Thank you, sir.
PP9.
Yes, sir. Yes, sir. Yes.
And you will be presenting microalgal diversity of Ajodhya Hills, Purulia.
Yes, sir.
Yes. Okay.
Yes, sir. Am I audible?
Yes, audible, and screen is visible also.
Yes, sir. So, good afternoon, everyone. I am Anupam Kundu, a PhD scholar of Vishwabharati University. My topic of today's presentation is microalgal diversity of Ajodhya Hills, Purulia. In this work, I have studied microalgal diversity of Ajodhya Hills. Okay, sir. Am I audible, sir? Hello?
Yes, you are audible. Yes, you are audible.
Yes, sir. Okay. In this work, I have studied microalgal diversity of Ajanta Hills in Purulia, West Bengal. First of all, let me tell about my study area, Ajanta Hills. So Ajanta Hills are small plateau with hilly surroundings where rocks are made up of granite and gneiss rock. Here in summer, highest temperature rise up to 45 to 47 degrees centigrade. Though Ajanta Hills are very highly biodiversity rich area, but there is not a single report documented on algal biodiversity, and therefore I selected this area, this study area, to explore microalgal diversity and their bioactive potential. At first of my work, I have collected crust samples. I've collected crust samples from rock surface, rock wall, tree trunk, waterfalls located there, and then collected samples were inoculated on agar plate containing BG11 media, and after that, pure cultures were made using several types of subculturing, and later photographs were taken using trinocular light microscope and documented using monographs and different literature. Then Bayesian method of phylogenetic tree was also made using nucleotide data to study the evolution relationship among them. And after that, metagenomic study was also done for unculturable microorganism from environmental samples there, and their genomic DNA was extracted, and 16 sRDNA metagenomics gene sequencing was also done for metagenomic analysis. And come to the next topic. My results, you can see my result. There are, though there are many of organisms are there, but there are a total number of 24 microalgal strains were isolated, identified, and documented here. Though there are also some other organisms, more than 1,000 of cyanobacterial and microalgal species are also present there. Out of them, 16 were isolated from rock surface of hills, and two species were isolated from nostoc, and nostoc are two species from which I isolated from tree trunk, and 6 species were isolated from waterfalls. I have also found some other species which are not identified yet. So I have not here present here them. And finally, the chronograph portion, you can see chronograph of metagenomic study. You can see showed that nostrils are most abundant with 51%, and whereas other organisms like Cytonema microcolia, Cytonema 5%, microcolia 3%, and Bacilloma 1%, Oscillatoria 2%, and Sinococcus 5%, Crucoccus 1%, and others, many species are present here. And lastly, I want to thank my guide, Dr. Gyanendra Roth, and co-guide Sika Mondal. And I am also thankful to organizing committee for giving me this opportunity to present my works. And in future, I want to work with some metabolites. I will purify and isolate some metabolites and will check their bioactive potential. And thank you.
Thank you, Anupam.
Yes, sir.
Any question from the audience, speakers? And we are very glad that we have Professor Andrzej Witkowski from Poland. So welcome, sir. You also may ask question to our last presenter. I think no question
No
for
question.
Anupam. Yes. So thank you. Oh, any questions, sir?
No,
Professor Witkowski?
I'm
question?
fine. I'm fine.
Okay, okay. So thank you.
Okay, thank you all participants. So all have given a very good presentation, I will say. Very good presentation. And hope they will do good work in future.
And all participants are requested to follow the guidelines or the suggestions. Thank you all. And I may request Dr. Nilupar Khatun may proceed, please.
Thank you, Gaur. So as we move on to our next vital lecture that we have, we are very glad and fortunate to have amongst us Professor Andrzej Witkowski, University of Szczecin, Poland. And
Hello.
who would better than Pia Roy Chaudhary to introduce him. So I will give over to Pia to please introduce her. Pia, are you there?
Yes. Can
She's
you hear
also from
me?
University of Szczecin, Poland. Yes, Pia, you're audible. Please go ahead.
It's my great pleasure to introduce Professor Andrzej Witkowski to you all. He is a renowned datamologist and senior professor of the Institute of Marine and Environmental Science, University of Szczecin, Poland. He has been awarded his PhD in geosciences from University of Warsaw and habilitated doctor from University of Gdansk. He has been awarded many prestigious fellowships like Dept Fellowship, Scholarship of Swedish Institute, Scholarship of Humboldt Foundation to pursue his research work in various countries like Germany, Sweden, Denmark, England, Indonesia, Vietnam, etc. He is working in this field since last 40 years. He has developed datum culture collection unit in University of Szczecin, where he has datum culture collection from all over the world. He has more than 200 publications and over 6,000 citations, and he has written multiple monographs. For this monograph, he is very familiar to all phycologists, and for his monograph on identification and classification of marine datum, he has been awarded by West Pomeranian Nobel Award. We are very glad to have you as a speaker, Professor, and this session is going to be very, very interesting. Over to you, Professor.
Thank you. Thank you, the organizers, and thank you, Pia. Good afternoon, actually, to everybody. Instead of saying he, I would suggest Pia to change it to we, because it's a teamwork and the achievements are done by the team and all young people. So I'm like a dinosaur, and my young fellows are very hardworking people, and I enjoy it. In fact, I spent my life with diatoms and mostly marine diatoms, and would like to show you today something what I would say I love in my scientific and my professional life, the marine diatoms, the diversity, the species, the genera, the species, and their geography. If I can show my, I guess this is my yes, this is my presentation. So can I start with a presentation?
Yes, you can start.
Thank
you.
You
Can start.
So I would like to say about the new, relatively new development in diatoms, use of molecular markers. This is interesting because, as you can see, it's quite international team. There was more young people working with us, but I just keep here those who contributed the most to this research. And we have been happy. I see, I didn't make it full screen. Yeah, is it? Yeah, okay. So this was the project we got years ago, 2012, but actually with this project all started. So we have started culturing diatoms. We have prestigious and famous scientists like Professor David Mann, Rosa Trabajo, Alistair Seddon, Evgeny Gusev, Thomas Bornmann, Catherine Riogobin, and John Bemiasa from Madagascar, who unfortunately passed away. And we have the PhD students, we have technicians, and one postdoc. That was the very beginning. So what was the project for? That was the principle was the biodiversity. How many diatoms, species, the taxa can we identify on the oceanic coasts in this littoral zone? Imagine sometimes colleague diatomologist fellow is asking me, how many samples would you estimate in a area like European tidal floods? And who can answer this question? I guess no one. So we've been thinking together, and we came to the conclusion it could be about 10,000 species altogether in the littoral zone of the European tidal flood. Then when the use of the molecular approach became more or less, I would still say, less the standard, then we decided to use traditional way. That means cleaning diatoms, making slides, and extracting molecular markers. The three which we selected are the most commonly used. So SSU, PSBC, and RBCL. Then the key point to have the samples, examination of the biogeography of littoral diatoms in various scales, in the local or the microhabitat scale, in the local scale, in the regional scale, and in the climate zone scale, and in the global scale. So we made a real, I believe it was a great success to work on the regional scale. I can point out two places. One is the coast of the South Africa, and to the lesser extent, the coast of Namibia. And the other one was Yellow Sea, Korea, and Korean, or rather opposite, Chinese and Korean coasts of this little sea. Imagine the extent, the length of the coast of South Africa is like 6,000 kilometers. When we include the Namibian coast, is getting even longer. And here I suppose in the Yellow Sea we have like 1,000 kilometers perhaps. So I'm going to show our study results from this area, from this little sea, the Yellow Sea. You can see particular sites like Laizhou, Changdao Islands, Yantai coastal sites, and Qingdao sites. It is strange, but still Qingdao is Yellow Sea. Qingdao is interesting because this is the powerful site, like 7-8 million people, inhabitants, and one of the largest Chinese oceanographic institutions. And then we have Dalian on the other side. For this, we had to use the ferry and go there and sample. And so we did sample from the external side and from the internal side of the Yellow Sea. Here you can see a more detailed picture, like Yantai, which is only 1 million inhabitants. I'm just not putting too much attention to this question of a number of inhabitants, because I remember, I'm aware that I'm speaking to our colleagues from India, and the population is not less, even if not more than in China. But here you, oh, I'm sorry, it doesn't work, or maybe it works. So what we did sample, please see, everywhere in area of Yantai, there is aquaculture. It's scallops, it's bivalves, it's shrimps, it's fish. Everything is culturable or viable and is in aquaculture. Here I was sampling, and the lady was interested in what I am doing here, why I'm climbing with the tide flow to her pond, where she is collecting as a member of the aquaculture team this bivalves. So I have explained her, and she was trying to make a drawing of this. And imagine in 2013 was this image taken. It's April. In 2015, these aquaculture didn't exist. The area was reclaimed, and there was no aquaculture, no shrimps, no bivalves, nothing. So we went to the open coast, to the sea coast, and you can see there is lots of diatom staining on the sediment. We have collected this, and this is the next site. This is the Changdao sites with very interesting diatoms. I was so fascinated with this girl, just a stranger. We have been sampling. She was playing there some kind of gymnastics, that I took this photo and I use it as a highlight of the Changdao sites. And again, as you can see, very rich, very fertile grounds. During the low tide, the sediment is greenish or maybe more brownish, and we sample. Then we went to the interior of the Yellow Sea, and we went to again to rich and fertile grounds with my colleagues from Yantao, from Yantai. It's Yantai Institute for Coastal Zone Research. We went for sampling, and over there you can see many boats ready for catching shrimps, fish, and so on and so on. And here is the coast. And we moved to the Dalian, where we sampled in this amazing situation. It looks like it's a low tide, but there is plenty of water. And this site was very interesting in general diatoms. This is Qingdao, which is the place I mentioned, where the big Oceanography Institute exists. And excuse me, I mixed. It was the Qingdao. So this is, I'm sorry, this is Qingdao. These are Qingdao sites. And this is Dalian. This is Dalian, the old harbor, and Dalian, the internal side of the, yes, very interesting issue where we have been sampling. There was a massive bloom of sargassum, one of the seaweeds, invasive seaweeds. You can see people voluntarily were sampling this sargassum and sending it or delivering it to the place where it was transformed into the nutrients. And people, this is not diatomologists, this is a low tide situation, and people are collecting their lunch or their dinner, just the, not the aquaculture, it's from the wild state. And again, you can see here the very fine sediment is covered with diatom stains, but below the sediment is apparently anoxic. Then we have the Korean side, the tidal flood of west coast of South Korea, which I sampled in November 2015. And it was very new for me. I've never been before in Korea. The colleague from Korea, Jong-gyu Park, Professor Jong-gyu Park, invited me to help his students and PhD students in consulting the identification of diatoms. So you see again this diatom stain on the sediment, and it is not only sand, not only mud. We also collected the small stones, and similarly to Chinese sites, there was lots of, or plenty of diatoms, but the culture from Korean sites was largely done by our Korean colleagues, and we published the results later together. And some results, even if I was aware how much diverse is the Chinese material, some results from Korea have been astonishing and remarkable for the global scale, I would say. To have the results valid, we made the measurements. We made also some formal action. Like for each site, we must have the GPS data, longitude and latitude. We measure the temperature, conductivity, salinity. Then we measure dissolved and saturation of the oxygen, and we measured pH. So you can see the pH is just normal oceanic. The oxygen content is moderate or maybe a bit low, but in some other places is high, and the saturation is depending on how turbulent water was there. But the salinity is almost like only marine. It's if it is 24 PSU, the border between marine and brackish water, then we are close to this value. So the material in China, collected in China, was brought to the lab in Szczecin. It was treated, and we call it from being wild to the monoculture. So the wild samples have been enriched with F2 medium. Then the isolation was performed with the Pasteur pipettes, glass Pasteur pipettes, which we work on. Many labs are buying very expensive microscopes due to some reasonable approach from a colleague who is selling this kind of equipment. We bought, I would say, very cheap microscopes, and we are happy with them and use them already 10 years and do not complain. So this is no name microscope, but originally I guess it was something like Olympus produced in China. And the monoculture establishment, it is time consuming. It takes re-inoculation and cleaning and growth in the chamber growth quite a long time, a few weeks, a few months. Sometimes it's forgotten and found back, like our colleagues know, it happens long after it was forgotten, and it is still viable. Then the harvesting of DNA and the strain goes into the culture collection, is kept. Many of the strains we keep for longer time die, but many are kept and are doing well in our culture, in our culture collection. I'm trying to remove this, but cannot. Okay, so once we arrive at the monoculture, imagine the Chinese material gave us about between 150 and 200 strains. One, we have the strain, the monoculture. The workflow is splitting into taxonomic and systematic survey based on light microscopy and scanning electron microscopy, and on phylogenetic survey, which involves DNA isolation, PCR reaction, and the molecular marker sequencing. I've told you we use these three markers. But it doesn't mean there is no more elaborate work in our lab, because practically each of our postdocs and each of our PhD students can work on genomic material, either mitochondrial and or plastidic, and it is curated or supervised by Dr. Romain Gastineau. Phylogenetic analysis involves maximum likelihood or Bayesian or parsimony, and then we arrive at the phylogenetic tree, which is the essence, I would say. There are different opinions. Some colleagues, even with whom we are working, say, do not put too much attention to the molecular markers. The other colleagues say it is the key point. I would say I am giving high value to the sequenced markers. Then in parallel, we work on chemical treatment of the material, wild material. We use hydrochloric acid and hydrogen peroxide. Why not strong acids? Because of our health. So prefer to work a few days longer than have the samples, the slides done quick, and suffer from pneumonial problems afterwards. So after a series of treatment, boiling or cooking, whatever however, it doesn't matter how you call it, we wash it, and then we have the microscopic slides. Then I personally love working, doing pictures. It doesn't matter whether it's live material, whether it's dead material, and I love SAM and FIB, focus ion beam. It's not the tool which is frequently used for diatoms, but it is. So you can see this is cooking. It's dropping the material for the slides. Then we have slides. At present, I guess it's much over 28 diatom slides. Each slide is in parallel. In our culture collection, we use high resolution microscopic setups. And this is Nikon, but we also use Zeiss, and that is unfortunate. We use someone's electron microscopy, scanning electron microscopy. We use either in our town in Szczecin at the other University of Technology, or we use it as a guests in Warsaw, in Rzeszów. This is making diatoms beautiful, and the photos are spectacular. So the variety of plastids and SAM, the variety of shapes and symmetries and so on. It makes all thousands, maybe dozens of thousands of images we have. It was not so easy in the former century. We had to use the negative films. Now we make thousands of digital images, and we keep them. And this is something what I mentioned a while ago, focus ion beam. It's the machine which allows to mill the diatom and cut it. So you can see, you can cut the diatom by this way. You can forward with the next cut. The cut, it's not cut, it's milling. And the milling agent is gallium ion beam. And you see it's continued. It's, of course, it is irreversible. If once we cut the diatoms, we mill the diatom, it is destructed forever. So once we have the molecular markers, we have the process of concatenation is done. And as I said, in the world where everybody is trying to be quick, fast, we use this 3-gene phylogenetic trees because the resolution is better, support is higher, and there is little unresolved situation. So more work, more effort, more time, but seems the effect is much better. This is our tree we made with 34 strains from the Yellow Sea coast of China. So we have some strains in Surirelatse, in Catenulatse, many strains. And it's funny because, funny is not the right word. It's interesting that most of these grown strains were new species, and there was also quite a number of new genera. All what is bolded here, you can see, is meaning it's a new species, and all definitely marine, but some you see still I would say over 150 strains are just touched, are not properly studied. Maybe 20 was in the tiles, and the rest is waiting as usual, better time in science. So as I said, we start with the light microscopy. So we have, you see, Nitzschia here, we have Diplonase, Cocconase, we have Nitzschia, Treblionella, we have Oryzaformis, Navicula, and some Catenulatse, some Surirella, Calonase, and Nitzschia. On the other plate, we have again Halamphora, Nitzschia, and this is a new genus, this one, which is Sternimirus. This is very interesting diatom. It's Amphora vix visibilis, which is placing itself in on the tree as one of the basal strains among the Raphid diatoms. And here you see also big Pleurasigma, which is quite strange. And as I said, imaging living diatoms makes fun, and we do this. So in fact, quite soon I spotted that we have not only local taxa, but in our hands we have some taxa which we can say cosmopolitan, ubiquistic, and this was the example. Nitzschia volvendirostrata was an example of ubiquistic species taxon. We found it in Yellow Sea in Yantai. We found it in Mozambique, in also Red Sea. And here you can see. No, sorry, it's not this one. It is oh. So it didn't fit, so it must be 36. I'll move with this. So this is the Red Sea, Saudi Arabia. So this is Nitzschia volvendirostrata. Sorry, the other one is Nitzschia tracheiformis, which it was like a Pandora box with Nitzschia, with this number of sampling sites and strains from, you see, New Zealand, Australia, up to Southeast Asia, Red Sea, South Africa, which I mentioned, our best studied site, Madagascar, and Europe all over, and the Baltic Sea, and the Arctic, and America, even the French Polynesia. It was not difficult to get this variable strains, and something what we used to say, Nitzschia dissipate, Nitzschia, strange Nitzschia. And here you can also see amphora, which I mentioned a while ago. So this is Eunotia and Araphid diatoms, and this is the Bacillariaceae. Again, images of our Nitzschia volvendirostrata. But Nitzschia tracheiformis turned out to be a very rich complex, very rich in species. So this is the pardon, original strains isolated from the Yellow Sea, and it looks like this. But then Marta sampling in Turkey, in Croatia, in other places, in Tsitsikama, in the Barents Sea, Corpus Christi, noted that we have everywhere, we have this, also in Korea, we have this strain. So it is like, as I said, a complex of species. And they need further research. But why it is so interesting? Because it's my guess or idea that among these diatoms belongs one of the domoic acid producing diatom, and actually it's the only Nitzschia, because as you know, it's only pseudonitzschia producing the toxic neurotoxin, which is domoic acid, and there have been some victims, some deaths after eating the bivalves after the bloom of pseudonitzschia in Canada in 1990s. So this is very interesting diatom, and it definitely it needs a more careful look and more effort. there is a problem with Nicia varyngiaca, Navis varyngiaca. Just I wouldn't say more formal. It's a technical problem because none of the 3 genes we sequence for the diatoms have been sequenced with Nicia Navis varyngiaca. It is only LSU sequence, so that's the problem. But as you see, many strains and a very rich material for comparisons. I put here the places where each of the strains, I don't mean it's one species, but at least here, maybe even here, it is from forming one nice clade. Even one of the turtle strains, Caretta, done by Marta, sampled by Marta and sequenced by Marta, shows this. Then we have one of the very cosmopolitan diatoms. I think it is a diatom which is number 1 or number 2 in terms of geographic range. It's Amphora helenensis. It was described from the temperate coasts of South Africa, from the south of the South Africa, southwest. That means it is the diatom which is impacted by water from Antarctic, flowing up north from Antarctica up to the system of Benguela. That's very nice because we spent so much time studying this area, and we studied the original material of Malcolm Giffen, which is also valuable. So then it turned out we have novel genera in the area because this diatom was resembling, I don't know whether the audience will agree with me or not, this diatom resembles rice grain, and this was my idea to name it Oryza formis, which means similar to the Oryza rice, Oryza sativa, which is just rice. But it was quite interesting that this species is occurring only in the marine environment, but only in the Holarctic realm. That means in northern hemisphere, in temperate water, marine waters of the temperate of the northern hemisphere. So you can see it's North Atlantic, it's temperate northern Pacific, and temperate northern Pacific in the west. So this is 1, this is 2. It's also found up north, and then we have it in California, in San Diego, where it was sampled by our colleague Matt Ashwood. So 3 strains and all grouping together and belonging in Plagiogrammaceae. Very, very interesting finding. But something what is involving us and very much, and with what we spend quite a lot of time, as a team, as a research team, when I speak this, I say this, I mean about our taxonomic work and phylogenetic work is Stauroneidaceae. Maybe less Cocconeidaceae, although we also do some, but Stauroneidaceae is our let's say focus. So first we found Colbesia, which is monoraphid diatom, as you can see here, one valve with a raphe externally, internally, and one valve rapheless or with a sternum. Of course, it has the raphe when it's growing, but for a short time, and then the raphe is sealed off by the silica. Then we have some achievements in Astatiella. It was a big review work or research work by our team, guided by Eva Górecka, where we described a number of Astatiella, again belonging in terms of the molecular phylogeny, belonging in the Stauroneidaceae. We named this diatom Astatiella vangi, which is in honor of our colleague Inchou Wang from Yantai in China. So this is Astatiella vangi, but you can see, please see Schizostauron and Astatiella is still, there are some problems, of course, as is science, but it is interesting, and let's say we guideline the lines for this group of diatoms. Quite interesting, this diatom is Cocconaeus. Most people think it would be Cocconaeus neotumensis variety submarina, but it is not. It is, in terms of morphology, it looks more like Cocconaeus mascarenica. It's Indian Ocean diatom described by Dr. Riogobin, and we have it in Yellow Sea. From the culture, we have interesting diatoms. I would be not astonished if we have here 1, 2, and possibly 3 diatom, which are new for science, maybe new genus in this case. We'll see when we go further. And then we go to the Surirellaceae and Entomoneidaceae, where we had just 2 strains. I shown LM images of this. I think Entomonaeus is a new species, and Surirella resembles Surirella fasciculata, so it's interesting. On the Korean side, we have lots of Simonsenia. In the, let's say, in for instance, in Gulf of Mexico, we have 1 species. In the Baltic Sea, we have 1 species. In the Western Atlantic, we have 1 species. And in Korean coast, we have like out of 6 or 7 marine Simonsenia, we have 5 occurring. It's interesting why the evolution is promoting, benefiting this kind of morphology. It's a very special morphology, and it is, you can see the reef is raised, and its external view, its internal view, and you can also see it in transmission electron microscope, and here you can see the position on the tree. So it is Simonsenia. We described several species. Simonsenia medlinie, one of the recent description. And to use as a highlight, I would like to point your attention to another interesting phenomenon of Korean yellow sea, the new Proszkinia. Again, with the number of species, maybe 10, 12, more than half we have in one site in Korea, in the Yellow Sea, Korean Yellow Sea coast. You can see newly described species: Proszkinia staurospeciosa, Proszkinia luticola, Proszkinia impar, Proszkinia modesta. So, of course, I could list hundreds of species and genera, but we'll cease with this and try to sum up. I see the study we've done in the Yellow Sea Chinese and Korean sites, some kind of foundation for future metagenomic and environmental sequencing, possibly because many of these diatoms are found worldwide. I've seen already reports that this or this diatom we described from Yellow Sea are now found further worldwide. And this, of course, is curated. The DNA sequences we have in our possession. Some material is accessible in the partner institutes like Yantai or in China, or Gunsan in Korea. There is, you may have noticed that there is a big benefits. The largest benefit of these cultural facilities is the record of diatoms in littoral, marine littoral. I mean, the big diatoms we find also in microscope, but the smallest, like Oryza formis, like Sternimirus, it's a little chance that we will find them and be able to identify correctly. Of course, these big diatoms are dominant, and they do not escape our attention. But the tiny diatoms have no chance to be observed in processed material, and we've seen many such cases among diatoms which we have in the DNA data banks, like from Greenland and from South Africa, from China and from Western Indian Ocean, and so on and so on. And Western Indian Ocean, Red Sea, and China. That works perfectly. Many of the taxa which we do not publish yet. Looks are new to science. It's not only the species level, it's also the generic level. So we have 4 years to work. We can work years and publish this data, but it's not our aim, and it's not our focus. Our focus is to make it broader. So I estimate that in the Yellow Sea coast, the number of novel taxa at the generic and level species could be 30%. Several new genera, just I can say this and this and this, just only looking at the material we have, like Sternimirus, like Oryzophomis, and so on. Hope soon we will work on the diatoms from India, from the Indian Ocean in India, and enjoy and be happy with new records and new species and new genera. Thank you very much for your attention.
Thank you so much, sir, for indeed a very informative presentation. We're indeed very fortunate to have you amongst us. Now the session is open for interaction. If anyone has any question, please, you can drop in the chat box, or you can just ask yourself. Anyone from the audience?
Yes, Nilofer, I would want to ask Professor Witkowski.
Yeah, please go ahead.
Good afternoon, sir.
Good afternoon.
This is afternoon for us. Probably it's something
also for us. Also
okay.
for us.
Okay. So I'm Dr. Neera Shincharkar from Kalyani University, and I've been working on the diatoms of Sundarbans for a considerable period of time. And I just had one question in mind. That is, we have, certain molecular level data. A lot of analysis was done, and a lot of SEM, the micrographs are available with us of a number of species from Sundarbans, from the mangrove areas of Sundarbans. We had kind of, we were successful in establishing their cultures also at one point of time. And then the pandemic came in. So what happened is that we lost most of our cultures during that time, but we still have with us the molecular data and the SEM. Sir, could you, kind of guide us onto how, because we are kind of sure, me and my team are kind of sure that we have, a number of novel taxa from this area. So looking into the molecular data, of course, and the differences in the SEM of, the way we have the diagrams with us, the micrographs with us. So how do we proceed with, confirming or the publication of new species from this kind of a data? Because we have tried after the pandemic to, kind of get those species again in culture, but failed at times. Is there any suggestion that you could give me?
Yes, make the plates for each strain. You have the vouchers, right?
Yeah,
the light microscopic images. Make the include together, put together for each strain, light microscopic and electron microscopic images.
Yes.
That would be the very beginning, and we can see what you have in your possession. And then if you have DNA, you have to perform PCR and sequence them. If you have the sequences, we can help you. I mean, my colleagues can help you in aligning them and producing, or can guide you how to make the phylogenetic tree.
Sir, what I was asking you is that I don't have the organism in hand. Like I have to submit a voucher specimen of the organism, no? So I don't have the specimens in hand because the cultures were lost during the pandemic.
Oh, so then you can't describe it. If you don't have the specimen, then the type, the holotype will not be recognized. It must be physical image of the or voucher, but the image is the condition to have. Otherwise, it will be not acknowledged.
Okay. So with the molecular data and the SEM images, I cannot proceed with publishing it as a new.
No, with the physical specimen, you can't. Okay. Unless you confirm that you have the species which is already known, right?
Pardon, sir, I couldn't get you.
Unless you will confirm through the sequencing that it is the species which is already known, the established species.
Okay.
So that only adds your record to the pool of occurrences, right?
I get your point.
Okay.
Thank you, sir.
Thank you.
And how could we, kind of proceed with increasing the amount of culture material that we have? Like it's scaling up the cultures has become a very difficult. We can isolate, we can, put them into cultures, we bring them into cultures. We have a lot of established diatom cultures also in our lab. But the only problem that we are facing is that we can't scale up the whole process, like for other purposes.
It's the culture condition that you have to regulate in your own how to keep them, maintain the culture. That's doable.
That's doable. Okay.
Yes. Okay.
Thank you, sir.
Thank you very much.
Thank you. Anyone else who wants to, interact with sir or have any doubts? Anyone else from the participants? I think they have understood it. Anyway, sir, thank you once again from the bottom of the heart for giving your time, precious time. Thank you so much. We are fortunate enough to have you amongst us. Thank you.
Bye-bye.
Bye-bye. Thank you, sir. So with this, I would request now Dr. Goh to please announce the winner of the poster session. Over to you, Goh.
Thank you, Dr. Khatun. Now we got the results. So there are 3 awards for posters, first, second, and third, as declared. And first poster goes to PP10, and that is Orko Mondol and Ganendra Roth for phycoremediation potential of Alinostock SP for removal of arsenic from contaminated water from Vishwabharati. And second poster award, best paper award goes to PP7, that is Shashwati Bhattacharya and PP Borua from Guwahati University, presented freshwater algal diversity in some riverine ponds of central Brahmaputra Valley. And the third best paper award goes to PP2, that is Karishma Das and Bhavatarani Panda from Utkal University, who presented algicidal activity of associated bacteria Alkaligens aquatilis on Lingvia astuani and its effect on growth and cell viability of cyanobacteria. So congratulations to all of you, and thank you, Dr. Khatun. Now I may request her to give vote of thanks.
Thank you, Gaur, and a very congratulations to all the winners. So as we have come to the end of a fruitful webinar, as we know, this foundation was established to encourage and enlighten student researchers and young faculties. We hope that today's webinar have achieved the same. Thank you everyone, our respected speakers, participants, and everyone involved for putting up this webinar. Thank you once again. Stay happy and healthy. And at the end, as we say goodbye, we have a beautiful dance by Iman Day. Hope you enjoy it.
এসো হে ঝনক ঝঙ্কারে উড়ায় শঙ্কর খুলেছে দুয়ার দেরি নাহি আর এসো 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 সুন্দর এসো হে সমারোহে এসো হে পরমতর সুন্দর এসো হে
Thank you, Mon, for that beautiful dance. And with this, we come to an end. Thank you, everyone, for staying with us. Just an important announcement. All the participants will get the certificate within 7 working days. Thank you, everyone. Till we meet again. Thank you so much.